Whether you're a bioprocess engineer hunting for the perfect scaled-down model, a biotech founder architecting the leap to commercialization, or a scientific analyst who craves raw technical truth, you've reached the technical edge. In this space, we live by a single mantra: the process is the product. Welcome to the Biomanufacturing and Fermentation Technology podcast with Prasad Ernala. Hit subscribe and ring the bell to join a global community and stay at the vanguard of biotech translation. Let's build the bioindustrial future together.
Welcome to the debate. Today, we're opening up the lid of the bioreactor to stare into the abyss that separates a successful lab experiment from a profitable industrial process. We're dissecting a massive and frankly expensive problem in bioprocessing, the scale-up gap. The fundamental question is simple but, well, painful. Why does a strain that behaves perfectly in a 2-liter glass vessel often just fall apart when you put it in a 10,000-liter steel tank?
Right, it's the classic valley of death for biotechnology, isn't it? You have a pristine process on the bench, flat lines, perfect control, beautiful curves, and then you move to production and yields crash, cells die, and you're left just wondering what on earth went wrong. And the standard engineering answer is usually to blame the controls. Just clamp down harder, reduce the noise, and, force the massive tank to behave exactly like the small one.
But that's exactly the mindset we're challenging today. Our central question is this: when we optimize a bioprocess, should we be aiming for that perfect static stability, treating the cell as, a passive chemical catalyst that works best when nothing changes? Or do we need to fundamentally shift our paradigm and actually program dynamic trajectories? Should we intentionally use stress and fluctuations to align with the cell's own decision-making logic?
It's a question of philosophy as much as it is engineering. We're really debating the definition of control.
I'm the advocate, and I argue for dynamic trajectories. I believe the industry's obsession with static set points, keeping pH and dissolved oxygen perfectly flat, is blinding us to biological reality. Cells are active agents. To get high performance at scale, we must treat stress as a design variable and program the environment over time.
I'm the dissenter, and I stand for stability and minimization. While biology is, obviously complex, industrial manufacturing requires consistency. Intentionally introducing perturbations like gradients, oscillations, stress spikes, it just adds chaos to a system we're barely controlling as it is. The gold standard has to remain a stress-free, stable environment to maximize the catalytic potential of the cell without, burning it out.
Okay, I understand the appeal of stability. It feels safe. It's how we design chemical plants. But let me start by attacking the core assumption of your position. Traditional fermentation treats the cell like a bag of enzymes. We assume that if we hold the pH at 6.8 and the temperature at 37 degrees, the cell will just sit there and churn out product at a constant rate forever. But that's effectively treating a living system like a passive ingredient.
It's a simplification, certainly, but engineering relies on simplifications. If I treat the cell as a black box that converts sugar to product, I can design a PID controller around it. If I have to worry about the feelings of every single bacterium, I can't build a factory. Complexity is the enemy of reliability.
But ignoring the black box is why the factory fails. In a small vessel, or a large one, every single E. coli, bacillus, or yeast cell is running a continuous, sophisticated internal control algorithm. It's not passive. It's sensing. It detects a pH excursion. It notices a dip in dissolved oxygen, or DO. It feels a pulse of sugar. It integrates all that data with its metabolic history. And then, crucially, it makes a decision.
You're anthropomorphizing the chemistry a little bit there. When you say decision, you really just mean a shift in metabolic flux based on thermodynamics and kinetics.
A resource allocation decision. The cell has a limited budget of ATP, energy, and ribosomes, the machinery that makes proteins. It has to decide, do I prioritize growth? Do I prioritize repairing my cell wall? Or do I prioritize the thing you want, the product? My argument is that we need to stop asking what steady set points are best and start asking what temporal pattern, what program leads cells to choose the high production state.
Okay, I don't dispute that the cell has internal logic. Any biologist knows that. But I come at it from the perspective of efficiency. If the cell is constantly deciding between growth, repair, and production, isn't it our job to remove the need for repair? When you start talking about programming dynamics, you're usually advocating for what I call hard pushing. Early strong induction, high shear forces, fluctuating feeds. We know where that leads. It shifts populations toward survival modes.
Sometimes survival mode is where the productivity is.
But at what cost? If you trigger those survival modes, say the heat shock response or the SOS response in bacteria, you are diverting ATP and reducing power, like NADH, away from my product. You're burning expensive carbon just to keep the machinery running. The traditional objective, maintaining a stress-free condition, remains the logical baseline because it avoids creating damaged, non-producing cells. I am very skeptical about abandoning the ideal constant profile in favor of complex, bursty dynamics just because that happens at scale. We should be trying to fix the scale-up, not break the lab model.
I think that's the trap, though. You call it breaking the lab model, but I call it making the lab model honest. Let's look at the hidden control system. You can have 2 fermentation runs with identical end conditions, same final biomass, same residual sugar. But they can have vastly different productivities. Why? Because of their trajectory. Cells encode the history of perturbations.
But in small scale, say 1 to 5 liters, mixing is incredibly fast. Gradients are modest. Static optimization works there because the environment is actually uniform. So if I can get a high titer with a flat profile in the lab, that proves the biology is capable of it. Why would I complicate the small-scale model with dynamics that don't naturally exist there?
Because that static optimization acts as a false positive. It creates a strain or a process that is just fragile. It's like training a marathon runner on a treadmill with air conditioning and a TV, and then expecting them to win a trail race in the rain. Industrial tanks have spatial and temporal heterogeneity. You know this. Cells circulate through zones. They hit a high substrate zone near the feed pipe, then a low oxygen zone far from the impeller.
Sure, circulation times create oscillations. In a 100,000-liter tank, a cell might take 60 seconds to loop around, but we try to minimize that circulation time.
But you can't eliminate it, and those circulation cycles trigger stress modules. Osmotic stress, oxidative stress, proteotoxic stress. If your lab model is perfectly static, you are selecting for a strain that has never turned on those stress responses. Then you throw it into the big tank, it hits a gradient, it panics, and production crashes. The lab model must mimic this logic to be useful. We need to validate that the cell's internal control loop is aligned with the external reality.
Okay, I agree that scaled-down models should mimic industrial gradients to test for robustness. But there's a difference between mimicking a problem to see if a strain survives it and advocating for stress as a feature. You seem to be suggesting we should intentionally induce stress to improve performance, not just to test for failure.
That is exactly what I'm suggesting. I am saying we should weaponize stress.
And that's where I struggle. Stress is a penalty. If we look at single-cell RNA-seq data, which allows us to look at individual cells rather than the average, it's quite clear. Under stress, populations partition. You get a multimodal distribution. You have high producers, sure, but you also get a cohort of maintenance mode cells and a cohort of damaged non-producers.
But that partitioning happens anyway. Even in your so-called perfect reactor, population heterogeneity emerges.
But the goal should be to eliminate the stress that causes the partitioning. If I have a population where 30% of my cells are in survival mode because I pulse the feed or let the DO drop, that's 30.
Why would I design a process that encourages that? It's just inefficient.
Because stress-free is a myth, and it's also not necessarily optimal. You're equating happy cells with productive cells, but often high production is inherently a stress state. If the cell is happy, it wants to grow and divide. It doesn't want to make your monoclonal antibody or your industrial enzyme.
Production is a burden, yes, but we manage that burden by balancing the feed, not by shocking the cell.
Consider stress as a design variable. I'm not talking about uncontrolled chaos. I'm talking about specifying the amplitude and duration. For example, a controlled osmolality ramp or a small temperature shift.
Okay, give me a concrete mechanism. How does making the cell miserable help me?
It can induce
protective responses, chaperones, membrane stabilizers, protease inhibitors that actually support high-level expression. If you gently trigger the heat shock response, you increase the folding capacity of the cell. Then, when you induce your difficult-to-fold protein, the cell is ready. It has the infrastructure. If you keep them perfectly happy and then slam them with induction, they collapse because they haven't built up the tolerance. It's the difference between, say, inoculation and infection.
That's a compelling argument in theory, but have you considered the operational risk? If you miscalculate that program, if your temperature shift is 2 degrees too high, or your osmotic ramp is too fast, you push the system into a death state, you trigger lysis, you ruin a batch worth hundreds of thousands of dollars. Is it not safer to aim for stability and perhaps engineer the strain to handle the burden rather than, playing with fire in the process control?
Safer? Maybe, but less effective. We're leaving yield on the table, and we don't need a pilot plant to figure this out. We can do this in the 1 to 10 liter platform.
So what does that look like? You're proposing we don't run standard batch or fed batch anymore?
I'm proposing we stop doing what I call experiment 1, where we just try to find the best constant pH and simply watch the line go flat. I'm proposing we deliberately impose perturbations. We should be doing aeration reductions to mimic DO dips. We should use pulse feeds to create transient feast-famine cycles.
And how do you analyze that? Standard OD, optical density, and DCW, dry cell weight, are bulk readouts. They hide all these details. If I pulse the feed and the OD stays the same, I assume nothing happened. But inside, the cells might be screaming. You'd need a flotilla of omics data, transcriptomics, proteomics, to interpret every single run. That's not practical for a development lab.
That's a fair critique. The standard analytics are blind to this, but the solution isn't to avoid the experiment, it's to instrument for decision-relevant readouts.
Such as?
Off-gas analysis. It is criminal how underutilized this is at small scale. You need real-time oxygen and CO2 data. You can see metabolic shifts in the respiratory quotient, the RQ, seconds after a pulse. And you need to track stress markers, reactive oxygen species, ROS, or specific metabolite proxies. You need to map the decision timeline. When did the stress response activate? Did it recover?
I will concede that off-gas is vital. If you aren't measuring RQ or COER, the carbon evolution rate, you are flying blind. But let's get specific about the control strategy. You mentioned trajectories. One of the most critical relationships in fermentation is between specific productivity, we call that QP, and specific growth rate, or mu.
Yes, the QP versus mu mapping. This is the holy grail of dynamic control.
For our audience, let's just clarify. Specific growth rate, mu, is how fast the cells are dividing. Specific productivity, QP, is how much product each cell is making per hour. In a typical process, we just feed exponentially to hold a growth rate, or we do a linear feed and just let the growth rate decline naturally as the tank fills up.
Right, which is sloppy engineering. I propose an experiment 2 approach. You perform a dedicated characterization run where you step through different growth rates. You hold them at 0.1 per hour, then 0.2, then 0.3. You measure productivity at each step, and you will often find a peak, a specific mu QP max, where the cells are most productive.
And usually that's not at the maximum growth rate. High growth often triggers the Crabtree effect in yeast or overflow metabolism in bacteria, where the cell just wastes carbon making acetate or ethanol instead of your product.
Exactly. It's often a sweet spot in the middle. Once you know that spot, you don't just run a generic recipe. You design a dynamic feeding strategy, maybe an exponential feed or a diostat that forces the culture to ride that specific growth rate curve. You align the reactor control with the cell's physiological optimum.
That I can support. That is rational engineering based on characterization. But it requires a shift in how we feed. It moves us away from simple linear feeds. However, my concern returns to the expression burden. You're optimizing the growth rate, but the protein production itself is a massive metabolic drain.
It is. Continuous overexpression creates a metabolic drain that competes with vital cellular functions. It's like trying to sprint while carrying a heavy backpack. Eventually, you stumble.
Which brings me back to stability. If you have a constitutive promoter or you induce and just leave it on, the cell eventually burns out.
The stability approach argues for finding a level of induction that's sustainable, low and slow, like a marathon pace.
Low and slow is one way, but dynamic expression modulation is better. Instead of constitutive expression, why not use on-off patterns or logical rules?
Logical rules? You mean like if-then statements for biology?
Yes, modern controllers can handle this. When oxygen uptake rate, OUR, drops below X, trigger a feed pulse, or start induction, but if the CO2 evolution drops too fast, indicating the cells are crashing, pause the inducer.
So you're suggesting a feedback loop where the cell's metabolic state is actually driving the controller.
Break to let the cells repair and replenish their ATP pools, then high expression again.
I suppose that acknowledges the partitioning issue I raised earlier. If you give them a break, you might prevent the population from shifting entirely into that damaged, non-productive state.
It is. It's essentially interval training for bacteria.
Exactly. You're managing the population heterogeneity. You're not trying to force a biological impossibility, infinite production with zero cost. You're negotiating with the cell. I'll give you a break if you give me a burst of product. It's an interesting concept. Consensus building here, we both acknowledge that the always-on, high-induction strategy is often a failure mode. Whether you solve that by finding a stable, lower set point, my preference, or by oscillating the conditions, your preference, the underlying biological reality is the same. The burden must be managed.
We agree on the problem. We just disagree on whether the solution is a straight line or a wave.
Fair enough, but I have to bring up the implementation cost. To run these program trajectories, you need significantly more structured protocols. You need better sensors, tighter feedback loops, and data scientists to interpret all the off-gas data. For a standard lab running 20 experiments a week, is this actually feasible?
It doesn't require new hardware. Most standard benchtop bioreactors, the 1 to 10 liter systems, have the capability to do cascades and if-then logic. It's a mindset shift, not a budget request. You don't need a supercomputer. You just need to stop setting agitation equals 500 RPM and start setting agitation equals a function of DO.
The mindset shift is the hard part. It requires treating the fermenter not just as a bucket for growing bugs, but as a scaled-down simulator.
Yes, and the payoff is huge. Improved space-time yield. You get more product per hour because you aren't wasting time in suboptimal states. And frankly, fewer mysterious crashes.
I will admit, looking at the data on mysterious crashes, they often resolve into identifiable interactions when you look closely. A crash isn't usually random. It's a collision between a feed pulse, a DO dip, and a stress response.
Exactly. If you track the trajectory, you see the crash coming 10 hours before it happens.
I'm willing to concede that avoiding extreme stress is key, and perhaps my definition of stability needs to expand to include stability of the physiological state, not just stability of the pH probe. If oscillating the feed keeps the physiology stable, then perhaps that is the true stability we need.
I like that framing. Physiological stability through environmental dynamic control. It sounds complex, but the organism is complex.
Don't push it too far. I still prefer a flat line if I can get it. Simplicity has its own quality in manufacturing. The more moving parts you have in a control strategy, the more things can break when the sensor drifts or the pump fails.
But you see my point. And there's one more benefit we haven't touched on, downstream processing.
Yes, cleaner harvest streams. That's a major point for the minimization camp. If you avoid extreme stress, if you don't push the cells to the brink of lysis, you get less debris, less DNA, less host cell protein contaminating the product. If your dynamic control stresses the cell too much, the purification team will hate you.
Agreed. But chronic stress, the kind you get when you try to force a static, high production state on the cell that wants to quit, that causes lysis too. Dynamic control, giving those recovery periods, can actually preserve viability longer. So you get a cleaner broth because you allow dynamics, not because you force stasis.
That is a hypothesis that needs testing, but it's a valid one.
So where do we land?
I remain cautious. I think the complexity of programming requires a deep understanding of the specific strain. If you try to program a trajectory without knowing your QP versus mu curve or without knowing your stress thresholds, you are just guessing. And guessing with dynamics is a recipe for disaster.
You create a roller coaster that flies off the tracks.
I agree. You cannot program what you haven't characterized. That's why Experiment 2, the mapping of specific productivity, is non-negotiable. You have to know the machine before you can race it.
I'll summarize my position this way. Cells are indeed decision makers. Ignoring that is perilous. However, the trajectory approach requires a level of precision that many labs are just not set up for. The goal should be to find the simplest robustness. If a dynamic profile adds 10% yield but doubles the failure rate due to complexity, it's not a win. Reliability is the currency of industry.
And I would counter that the simplest robustness is often an illusion at the bench scale. The perfect run is one that is properly programmed to cooperate with the cell's logic. We must move from recipes to trajectories. Yield, robustness, and scalability are coupled outcomes. If you fight the cell's logic with static set points, you lose. If you align with it, you win.
It is certainly a perspective that makes the 10-liter for Metro a much more interesting place to work.
It stops being a pot and starts being a computer.
A biological computer, which is the most frustrating kind. It never runs the same code twice.
Indeed. But that's the job. This perspective is particularly crucial for yeast and peptide-producing hosts, where expression burden is the central constraint, but it applies everywhere.
We encourage our listeners to look at their own data. Look at those perfect runs that failed. Look at the messy runs that succeeded. There might be a program hidden in the noise.
Precisely. There is more to explore in the material, and we hope this debate has given you a new lens to view your next fermentation run.
Until next time.
Thank you for listening to the debate.
Hi there, thank you for staying with me until the very end. If these technical insights help bridge the gap in your own process, please hit subscribe and make sure to hit the notification bell. Stay connected for our next deep dive into the realities of biomanufacturing. I am Prasad Ernala. Thanks for listening.